All right, we're moving on to our next learning target in our macromolecules and enzymes unit. That is learning target 4. Students will be able to identify the properties of enzymes and explain the role of enzymes in chemical reactions. So this whole unit is on macromolecules and enzymes. So now we're getting to that second big part of enzymes and really understanding how they work. Enzymes are biological catalysts. What that means is that they can speed up chemical reactions. They do that by decreasing the amount of activation energy that's needed, and in doing so can increase the reaction rate. And so if you look here, here we have a normal reaction occurring, and you can see here again the amount of energy needed to get that reaction going is called activation energy. And so this is the amount of activation energy that would be needed if we added an enzyme right here in this dotted line. Look what's happened. We've lowered the amount of activation energy needed for that reaction to take place. And so that allows our reactions to occur at temperatures that are appropriate to our bodies if we can lower that activation energy. So enzymes are catalysts for chemical reaction in living things. They speed up chemical reactions. They lower the activation energy needed. And why that is important is it's so that the reaction can take place at cellular temperatures. When you think about it, if you're in the lab room, and a lot of times we'll use heat, we'll start the Bunsen burner or we'll have a hot plate going to heat up something to get that reaction going. That's what we kind of use as our activation energy. Well, we can't heat up our body cells, because if you heat them up too much, you're going to destroy them. And if that kills your cells die, guess what? Yeah, you die. So by having an enzyme lower the amount of activation energy needed so that our chemical reactions can take place, now they can take place at our normal body temperature. Some other things about enzymes is, remember that they belong to the macromolecule proteins. Remember we talked about that as being one of their functions, as being an enzyme. Most of the time they end in this suffix ase. So if you look at amylase and protease and any of those that end in sucrase, lipase, all of those things are enzymes. They end in ase. Not all of them, but most of them do. Now, there are some things that can affect how well an enzyme works. Temperature is one of those things. pH is another, and then enzyme or substrate concentration, like how much of it is there. Okay, so with temperature, too high of a temperature can break the hydrogen bonds and destroy the enzyme. We call that denaturing. Okay, that we denature the protein, we denature the enzyme. And so obviously it won't work if it's all broken apart, it just doesn't function anymore. pH, most enzymes work best with a neutral pH. So if it gets too acidic, too low of a pH, or too basic, too high of a pH, that's going to destroy the enzyme and not make it work anymore. The exception to that would be pepsin. You have pepsin as an enzyme that's found in your stomach. Well, you also have hydrochloric acid in your stomach, which is very, very acidic, very low pH, like around a 2 or a 3. That actually helps activate the pepsin, that enzyme, to help break down your proteins. So interesting, but most enzymes work better at a neutral pH, closer to a 7. And then enzyme or substrate concentration. As we're going to find out in a minute here, how an enzyme works is that it attaches to a specific substrate. Okay? Once there's a spot on the enzyme called an active site, and that's what the substrate attaches to. Well, if you only have so much enzyme available, once those active sites are filled, it doesn't matter how much substrate you have. If you kept adding more and more substrate, it wouldn't matter because all of the enzymes have already been taken. And it's kind of like if you went to a dance and there were only so many dance partners there. Once they were all taken, it doesn't matter how many more people you add to the room to dance. You know, they're all taken. I know, that's probably very outdated in my book here because I'm old. So let's see how enzymes work. Enzymes are very specific. Each enzyme structure allows only certain reactants or substrates to bind to the enzyme. Okay? So a substrate is any reactant that the enzyme binds to. Okay? And your active site is the area on the enzyme where the substrate binds. So if you look right here, here is the active site, and here's another one here, okay? And you can see that these substrates here, in this case there's two reactants, one is going to bind to this active site. You can see it's going to fit in just like a nice little puzzle. And here's the other one that's going to fit onto this active site. And here is your enzyme down here. So you need to know that enzymes are very specific. If a different enzyme came along, these two substrates could not hook onto that different enzyme because their shape is very specific to that particular active site on that specific enzyme. So when you think about all the different chemical reactions that occur in our body, we have different enzymes that are playing a role in all of those different chemical reactions. So pretty amazing. That's why enzymes are so important. So the lock and key model illustrates how an enzyme functions. Sometimes people almost think of it even as pieces of a puzzle fitting together, but sometimes you'll hear it called the lock and key model. Substrates, again, are brought together on the active site of the enzyme, and this forms what's called the enzyme substrate complex. So again, here are your substrates or your reactants. They're going to fit to the active sites there on the enzyme. Once they are all hooked together, we call that the enzyme substrate complex. And what happens here is, this is when the actual chemical reaction can take place. The bonds can be broken and reformed. And you can see in this case, we started with two substrates, one here and one here, or in this case, two reactants. And once we form that enzyme substrate complex and those bonds got rearranged, now we have one product that's going to be released. Okay? So can you tell just by looking at this what kind of reaction that is? Would that have been a hydrolysis reaction or a dehydration synthesis? Remember what synthesis means. It means to build. When you take smaller molecules, join them together to make a larger molecule. And that's exactly what's happened here. So this would have been a dehydration synthesis reaction. Now, when we are thinking about our digestive system, we're going to be focusing on three major enzymes, and that is what we'll do in our lab if we're able to do our lab in class. Is look at three major types of enzymes, keeping in mind that we have tons of other enzymes in our body. But we're just going to focus on a few of them so you can understand how they work. One of those types of enzymes is amylase. Okay? Now, your saliva contains what's called salivary amylase. So called that because it's in your saliva. Okay? So what happens here is that amylase breaks down carbohydrates into simple sugars or monosaccharides in your mouth. So that's one of the first places where digestion occurs, is in your mouth. You start chewing to kind of break the pieces up, but the actual chemical digestion involves that enzyme amylase, salivary amylase, breaking down those carbohydrates into the subunits, monosaccharides, in your mouth. Next, another one we would like to look at is protease or pepsin. These are enzymes that break down proteins into amino acids in your stomach. Okay? Remember, your stomach is very acidic. It has hydrochloric acid in it. So not a lot of other enzymes function very well in your stomach, but that pepsin is actually kind of activated by those acids, and that helps it in breaking down proteins into its subunit amino acids in your stomach. And then a third one we'll do a little bit more research with here is lipase. Lipase, again, you notice that it ends at ASE. A lot of your enzymes do. Break down lipids, specifically fats, into fatty acids and glycerol in your small intestine. Okay? And so you can see up here, here's your glycerol molecule, and then you've got your 3 fatty acids. Those are the subunits of a fat molecule. Okay? So when we do our enzyme lab, if we're able to do it in lab this year, those are the 3 main enzymes that we like to do some research on, because it kind of shows you some of the different parts of where your digestive system is found, and just an example of 3 of the different enzymes that would be found in your body. Okay, so let's just do a little bit of practice before we finish up this PowerPoint. What are these different parts of the diagram below? Okay, again, if you look here, the blue structure here, they all look the same. This is actually your enzyme. Okay, and here's the enzyme again. And one thing I forgot to mention, not only are enzymes specific, but they are never changed or used up in a reaction. Notice how here's where it looks here. Here it still looks like this here. It still looks like this here. It's never changed or used up. What you start with is always what you end with. That's why you can use that same enzyme over and over again to do that same kind of reaction over and over again in your body. Now what we're trying to do here is hook this substance onto the enzyme. This is what we call your substrate. Remember, your substrate is what is going to hook on to the enzyme. Another name for your substrate is your reactant. Okay, where does it hook onto again? What are these spots here called? Those are the active sites. They are on the enzyme. They're the area on the enzyme where the substrate or reactant hooks onto. Once we have all of this together, okay, once we have all of these joined together, this is called the enzyme substrate complex. Okay? And then what do we have at the end here? What do we call these 2 parts? That would be your products. That's what's released here at the end. Now, what kind of reaction would this be? We started out with one large molecule. Once we formed the enzyme substrate complex, that enzyme did its job. In this case, it's rearranging, breaking the bonds, forming new ones to actually break apart this large molecule into 2 smaller molecules. So this would be an example of a hydrolysis reaction. Remember, lysis means to break apart. So this one would have been an example of a hydrolysis reaction. So make sure you know how enzymes work, some characteristics of